Okay, this program is a review for Exam 1, covers chapters 1 through 3 and the associated lectures. Just a reminder, Exam 1 is 100 points. It'll be more than half the points will come from multiple choice questions. Okay, about a quarter of the points will come from an essay that you write. And then the rest is fill in the blank and short answer questions. This is, there's about, 10 points per chapter quiz, about 10 to 15 points per lab. Exams are the primary way that makes up your grade. Not your whole grade, but more than half. And so it's important to study for the exams and so that you get the most out of the course. With anything, it's repetition. When you're trying to remember or learn anything, repetition is the most important thing. And the number of times and the amount of time that you spent will be the primary determinant of how well you do. And so the best way to study is what's called distributed review, which just means to study repeatedly with intervals between. So to work on the chapters, to practice the questions, and then to repeat that again. Distributed review is better. Repeated short sessions are better than one long session. And so people do best studying 10 or 15 minutes at a crack, taking a short break, and then doing another 10 or 15 minutes at a crack. And you're most likely to remember if you use active methods. That doesn't mean reading or browsing a book or notes, but actually taking notes, handwritten notes, looking over old exams that you've already written, making review sheets, and quizzing by yourself or with partners where you write the answers down. Those are the best ways to study, the ways that will help you remember. So my advice is to plan, to plan how much you're going to study and to do it in short breaks, where you're taking short breaks. Okay. Hopefully everyone's read the chapters and done all the chapter quizzes, so you're in good position. You won't need too much more studying, but it's good now to spend a lot of time with those chapter summaries. Look at each of those bullet points, look at each of those words in bold. One of the main things is the vocabulary, the new words. What do they mean? And you can find those from the textbook, but you can also find them on the summaries from Moodle. Again, plan frequent, short study sessions. Rather than 1 4-hour session, it's better to make, three spaced out 1-hour sessions where you do 15-minute intervals and take 5-minute breaks between them. That's better. So this is a review that's designed to go over chapters 1 through 3. It doesn't have everything. It's not comprehensive. The textbook is comprehensive. But what it's designed to do is hit the highlights, to show what are the most important parts and to focus you on those. And so in chapter 1, that was our introduction. Who are the microbes? The microbes are, they're invisible invaders that cause disease, but they're also allies that help us do a lot of things. Okay, they fall into a couple groups. You should be able to name these groups, kind of name some features about them and give me an example. Okay, the bacteria are the organisms that are small. They have prokaryotic cells. They're too small to see with a microscope, really. They include things like E. coli and Staphylococcus. Some of them are found in our skin. Some of them are found in the environment. They can produce disease. Archaea are also prokaryotic cells, very small, but they are not generally a cause of disease. They're more common in the environment. They include things like methanogens or the thermophiles that might live at Yellowstone. Okay, and then we also have organisms that are eukaryotes, that are have cells like ours with a nucleus. Okay, these include single-celled organisms like amoeba and protista. It includes some algae, single-celled algae like diatoms. It includes fungi like Saccharomyces cerevisiae, the the yeast that we use to make bread rise and beer ferment, and also some animals that are microscopic, or at least have a microscopic stage. And then lastly, the viruses. Viruses aren't truly alive. They're chemical in nature, but when they invade cells, they can take cells over. Examples of those would include influenza, herpes, HIV, and of course, COVID-19. All right, another big thing on the organisms was that we have a scientific way of classifying those. We give each of those a 2-word name, the genus and species name, and that's Linnaeus that came up with that system. Okay, and so like Staphylococcus aureus is the name for a bacteria that causes infections and found in our skin and nasal cavity. Okay, we also classify organisms into larger groups where we base them on similarities. These include the domains, and there are 3 domains: bacteria, archaea, and eukarya. All right, and let's see. Then another big topic in chapter 1 was kind of what microorganisms do. What are the practical application? What is the scope of microbiology? Microbes are important in medicine as a cause of disease, but also to make cures for disease. We use them in pest control, where they're important in the environment, recycling nutrients, detoxifying wastes. They're important for agriculture, for biotechnology and genetics. We use them to make food, fermented foods in particular. So we should be able to kind of explain those. In chapter 2, we talked about how we can make the invisible visible. And so we talked about the history of the microscope, the light microscope, where we use lenses and we capture light rays and we refract those to make them larger, to get an image that's much larger. And this is basically magnifying glasses. And so talked about the different types of light microscopes. I'd like you to know bright field microscope and maybe something about like a phase or dark field, which are used to see live organisms. Okay, and there are limits to this. We talked about magnification, but we also talked about resolution. And we can only magnify somewhere around 1,000 times with a light microscope. To get higher magnification and better resolution, we have to go to electron microscopes. And these use electrons instead of light beams. Electrons travel in straighter lines, so you can magnify them more and you don't lose resolution. So we can magnify to maybe 100 or 200,000 times. Okay, this allows us to see most microorganisms. Okay, here's a scale, and microorganisms, bacteria, archaea, and eukaryotes are in the neighborhood of 1 to 10 UMs, which are micrometers, and the viruses are on the order level 10 or 100 times smaller. Okay, they can only be seen with electron microscopes. Okay, in this chapter we also talked about staining, how we prepare microorganisms to make them visible. We talked about lots of different types of stains, but the Gram stain was the one differential stain that I wanted you to know. This allows us to classify organisms on the basis of the structure of their cell wall. Some have a cell wall that makes them pick up and hold a crystal violet that will make them purple, and then others won't hold that crystal violet. They'll lose it when they're decolored, and we counterstain them with safranin so they'll appear pink or reddish. Okay. Throughout chapters 1, 2, and 3, we talked a little bit about the background of microbiology and how science works. That science is a way of learning about the environment, a way of knowing. It's based on observation, and then testing our ideas with experiments and using that evidence to argue about ideas. Using science, using observation and experiments, we can gradually get rid of wrong ideas. Okay, and this requires that we're able to discuss things. And so we want to have a fair argument, so that means you have to talk about what evidence you have, and you have to show it to other people and let them try and repeat it. So those are key things. And when we talked about different different guys here, and a lot of it was getting rid of wrong ideas, like ideas about what causes disease, whether it was miasmas or whether it was an imbalance of fluids or whether it was germs. Okay, and that took a long time to get the evidence that really clarified that, to get rid of ideas like spontaneous generation. And we gradually tried things and accumulated evidence to say, no, that's not the right idea. And as well, there's two guys there, Semmelweis and Lister, that gradually, really dramatically improved our ability to take care of people through recording the results of experiments on hand washing or using disinfectants. And so that history of ideas is also the development of science. And that's really kind of an important thing. What is the evidence, and how can we explain that evidence? And so we talked about in that as well, cell theory, the evidence that accumulated that all organisms are made out of cells. Life process occurs within cells, and cells come from pre-existing cells. And so those are big ideas that underlie microbiology. Okay. Then in chapter 3, we also talked about the different types of cells. And so that lots of that evidence is, using electron microscopes, and then it was chemical studies that we didn't talk about. But we are able to divide cells into two basic types based on their features. Prokaryotic cells, bacteria and archaea, are smaller cells and simple. They have relatively simple structure. They have a rigid cell wall made of peptidoglycan or pseudopeptidoglycan. Inside of that is a plasma membrane that controls what goes in and out. Then a fluid-filled chamber of cytoplasm that contains a nucleoid region that has DNA that's not separated by a membrane. These cells also contain 70S ribosomes. Okay? All cells, all prokaryotic cells have those features. Some have additional features like capsules and plasmids and inclusions, but many don't. Okay? So there are those basic features. Okay? Eukaryotic cells, on the other hand, are larger, much larger. The average eukaryotic cell is probably at least 10 microns in length, and in volume probably 50 or 100 times larger than a prokaryotic cell. All eukaryotic cells have a nucleus, a membranous structure that separates the chromosomes from the rest of the cell. Most of the DNA in eukaryotic cells is in the nucleus. Okay, all eukaryotic cells also have in their cytoplasm 80S ribosomes. It's a larger ribosome not found in any prokaryotes, and it might even be attached to an endomembrane system. They also contain a network of proteins called the cytoskeleton that's associated with movement and structure. They all contain a mitochondria where aerobic respiration takes place, again, surrounded by membranes that separate it from the rest of the cell. And that mitochondria contains a 70S ribosome, contains its own DNA, and is capable of reproducing on its own. And so that's part of that endosymbiosis network. The endomembrane system you find in most of those cells. Some cells have a chloroplast. Okay, there's lots more details on prokaryotic and eukaryotic cells, and some of those details might be on the exam, but know the core structures for sure. Those core structures will definitely show up, and I'll definitely test you on those. Okay, good luck, study hard, and I'll be at my office hours before the exam, so if you have any questions, you can show up on Zoom, and maybe I can answer them for you. Okay, good luck.